All right, gonna go over a couple things for class. I'm gonna be drawing on the whiteboard. First thing we're gonna go over, hopefully you can see that there. Perfect. Neuromuscular junction. I'm gonna go over kind of how those fire, how they travel down, how they release calcium out of the sarcoplasmic reticulum, thus leading to muscle contraction. Then we'll talk about relaxation and kind of how everything goes back to the way it needs to be so it's not in a contracted state. Then I'm going to go over muscle energy types, muscle cell energy types. We're going to talk about ATP. We're going to talk about different ways that we get energy within the cell and what those are good for and how long they last. So that'll be a good topic. The next thing we'll do is we'll go over smooth muscle. So with smooth muscle, I'm going to go over 3 different types. We have our type 1, type 2A, type 1 slow, type 1, type fast, type 2A, fast, type 2B, or type 2X. Okay? We're going to talk about different things with those in terms of mitochondria, thickness, blood vessels, and kind of important things that we need to know in regards to how smooth muscle functions and why it's different than skeletal muscle. Then I'm also going to talk about contraction with smooth muscle. They do contraction a little bit different. They have something called a lag phase where they can stay contracted for a long period of time without the use of ATP, and that uses MLCKP or MLCK. We'll talk about those enzymes and how they help regulate that for smooth muscles. And then we'll talk a little bit about fatigue, high frequency and low frequency. So we'll talk about those things as well. And then the last thing is I'll talk about muscle sensory receptors. So we'll talk about the extrafusal muscles, how there's intrafusal muscles that are proprioceptors. They kind of sense movement, and that's how you can see where your hand is when your eyes are closed. So we'll go over all those things, and with that we'll feed into those intrafusal muscle fibers, which are actually spindles. When you have a bunch of them together, we call those a spindle. And then we have a Golgi tendon, organs. And then we'll go over stretch, reflex, and withdraw, and kind of how they work together to keep our bones from splitting in half and keep our tendons strong. So I'll go over all that. Hopefully we can go through this pretty fast, but we'll start with the neuromuscular junction with the action potential and kind of explain how this is going to work for contraction for skeletal muscles. Okay, so first thing I want to draw here is a spinal cord, and this is almost like a transverse cut, so we're just cutting it right in half. And if we were to look down the spinal cord, we would see that there would be this horn looking structure, and then down the middle we would see a hole like that. So this is called our lateral horn, and this is on the ventral side. So we know that this has to do with motor, okay? So this is actually after a stimulus has been received. And we want to control a muscle, and we want to tell the muscle to contract or to relax, or we want it to do something. And so we're sending a signal down our spinal cord, and then it's going to come out a nerve called the alpha motor neuron. And that's going to come from the lateral horn here, okay? So we'll have these neurons synapsing here, and we're going to have these coming out like this, okay? And they're all going to feed into this. This is called our alpha motor neuron, okay? Alpha motor neuron. This is going to continue, and then I'm going to blow this up right here. Perfect. Okay? This is going to be our axon. Now, in previous videos and previous lecture information that we've gone over, we know that action potentials are occurring through this axon if we're trying to send this chemical message That will turn into an electrical message, which will then allow for the effector to do something that we want. What's going to happen is this is our axon. If we had an action potential that's firing, we had enough EPSPs build up here, we're sending an action potential. It's all or nothing, we're going. Action potential sends down, we know that we have these sodium voltage gated channels. That are opening up, okay? And when they do, because threshold has been exceeded, we're in this depolarized state, depolarization. We end up having positive charge continue to come down because we have so many sodium ions flooding in. It fills the cell positive, bringing that resting membrane potential up past that threshold, and we're at an action potential firing down the cell. Now this is important because as we get to here, this bulb end, we call this the axon terminal. And at the axon terminal, this is where we actually take this electrical signal and we turn it into a chemical signal through or via the release of neurotransmitters. How this works is we have this channel right here. And this is called a calcium voltage gated ion channel. Okay, a calcium voltage gated ion channel. What happens is if we get enough positive charge that comes in, the goal is to have this channel open up. When it does, calcium comes rushing into the cell like this. Within the cell, we have special vesicles. Okay, these circles are called vesicles. Okay, within these vesicles, we have these little dots representing NTs, or neurotransmitters. Okay, so we have vesicles, we have neurotransmitters. The reason calcium is so important is because when it floods in, we get all these little calcium ions within the cell, right? Their job is to help with the binding of these snare proteins that exist on both the vesicle and the membrane of this axon terminal. These are called V-snare proteins. And these are also on the membrane, and these are called T-snare proteins. And what happens is calcium, when it floods into the cell, it has this ability to come in and help these snare proteins bind. When they do bind, and I'm going to bring this over here, when they do bind, we have the vesicle that begins to start and fuse with the membrane, because those snare proteins are going to grab onto each other and they twist and they pull and pull and they pull this vesicle to the membrane. Therefore, we can have exocytosis of NT, neurotransmitters, right? So now these neurotransmitters can be released into the synaptic gap or the synaptic cleft. This region right here is called the synaptic cleft or gap. And this is where we have neurotransmitters floating, sending a signal to another receiving cell. Now what ends up happening is these neurotransmitters In this case, with the alpha motor neuron for skeletal muscle, happen to be acetylcholine. I'm going to label it ACH for short. Acetylcholine. This neurotransmitter, acetylcholine, will send a signal for us to further cause contraction. But I want to take a minute here and pause and explain something really quick that's important. And that is, this is an enzyme right here, okay? This right here is an enzyme. And what this enzyme does is it's called acetylcholinesterase. Acetylcholinesterase. And its job is once acetylcholine is done its job with binding to a ligand, it's going to clean up any acetylcholine floating inside us all, so we don't have continued contraction when we don't want it. So what ends up happening is it cuts it in half. We know that there's mitochondria within the cell as well. And that's where the acetyl-CoA is coming from. And then the choline gets snipped off. That comes from diet. And then we can recycle these parts and put them back in. Okay? And so that's what that enzyme, acetylcholinesterase, is doing, is helping to regulate the neurotransmitter acetylcholine that's within the synaptic gap, so we have controlled fine muscle movement when we want to move our muscles, okay? So I'm gonna draw something else over here. This is gonna look really ugly, but basically you have layers to your muscle. Okay, and then we have these right here. And if we were to pull one of these out, like this, we have that. So basically, this is a skeletal muscle, like a bicep. Okay, skeletal muscle like your bicep. And this outside membrane layer on the outside here, dotted, that's called the perimysium. Perimysium. No, that's not called the perimysium. That's called the endomysium. Then you have these other circles here, okay? If we go a layer down, so we have the muscle, right? The full muscle intact, and that's called the endomysium. Then within that, we have these bundles of muscle fibers. And these are called fascicles. Okay? Fascicles. So we have fascicles, and there's a thin layer, almost like a fascia. That's what this is, endomysium. It's a fascia. There's this thin layer around the fascicles called perimysium. Okay, perimysium. Then, beyond the perimysium, we then have this area right here. We know that the perimysium covers muscle fiber bundles. So multiple thousands of these muscle fibers, and they're called a fascicle. Within that is one muscle fiber right here. This would be a muscle fiber. Okay, this could be our muscle fiber right here. And with the muscle fiber, what ends up happening is, there we go. Yeah, with these muscle fibers, they actually are encased by a layer called the endomysium. And directly below that is called the sarcolemma, which is that phospholipid bilayer of the cell membrane, okay? And so if we were to continue this muscle fiber on, we know that the muscle fiber then is consistent of all these myofilaments. Okay, and that would be nebulin, myosin, actin, and titin. So we have these things called myofilaments, okay? And if I were to continue this, and let's pretend that this fiber we then expanded open right here, what we would see is we would have something like this occurring. Okay, can we see that? The lighting's terrible. How's that? Can we see that? Yeah, that's good. Okay, so we have, let's go back to where we were at. We have neurotransmitters that are being released here called acetylcholine. Acetylcholine. Now, there's a receptor on the membrane. This is called the sarcolemma. Okay? Sarcolemma. That means cell membrane of a muscle. Sarco, muscle, prefix for muscle. So we have a muscle, sarcolemma, muscle membrane. Okay? We have these channels that are found within this membrane. Okay? And what we call these are ligand gated ion channels. And so we have these neurotransmitters that will diffuse across the synaptic gap, and we have a receptor right here on this ligand gated ion channel. And this is called an N1 receptor. N1 receptor. The neurotransmitter binds over here. Okay? And this actually opens the ligand gated ion channel. So then we can have sodium rush into the cell. Now this causes a depolarization, and we call this end plate potential. Okay, end plate potential. Okay, that's called the end plate potential. What ends up happening is we have this positive charge that builds up within the cell. As this positive charge builds up within the cell, this starts to have a sphere of influence within the cell. And we start to notice propagation. So what we have are these sodium voltage gated ion channels that begin to open up. And we have sodium, which is in abundance, high concentration outside the cell. It wants to go from high to low. We know that this is called a concentration gradient, and so it's going to flood into the cell. Thus, we get this positive charge buildup within the cell. And this leads to propagation and continuation of this positive charge having an effect in depolarizing the cell, causing this action potential to occur. We get to a spot over here known as the DHP. Okay, DHP. This gets activated and opened up because of this positive charge that's flowing down. And what it does is it opens this receptor here on this cell known as RYR. And when this activates RYR, somehow it's able to open it up and allow calcium to flood out. Now this right here is called the sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. And this is called the sarcoplasmic reticulum because it's the endoplasmic reticulum in a muscle. So we add that word, sarco, meaning muscle. Okay? Then we have calcium. Now calcium's in high abundance over here at point of where we would leave because of proteins called calciquetrin. Now calciquetrin is going to keep all the calcium close to this opening spot by the RYR channel, so that way when it does open we have a high amount of calcium flooding out of the cell into what we call the sarcoplasm. Now one thing about this structure anatomically is this region right here, these are called terminal cisternae. And one thing I failed to mention is this membrane continues down. This is the ECF out here, and this is the ICF in here. And this is called a transverse tubule. Or T-tubule. And this is important because it allows us to get this signal down here by the sarcoplasmic reticulum, thus allowing this calcium to flood out of the cell. And into the sarcoplasm. Transverse T-tubules, okay? So the same thing's happening over here. This is same, okay? And so when we have both of these with a T-tubule, we call this a triad. Okay, we call it a triad. So we have a positive charge coming down. It opens up DHP. DHP gets activated, activates RYR. RYR opens up. Calciquetrin is holding calcium right next to the edge of the sarcoplasmic reticulum. Sarcoplasmic reticulum is high in calcium concentration. RYR opens, calcium floods out, and now it's in the sarcoplasm. It's in the sarcoplasm, and it's going to float over to where now we need it, and we need to know what it's doing now, and why this is so important, and why we spent so much time explaining this process of the neuromuscular junction. What's going to happen is I'm going to draw a sarcomere that's found within a muscle like this in a muscle fiber. And we have these Z lines here, and we call these Z discs. Okay, Z discs. Within the Z discs, we have a part of the cell called nebulin. And below that, we have this squiggly piece here called titin. And on nebulin, we have these circle structures, almost looks like a beaded necklace, coming around nebulin, wrapping around like a alpha helices. And this is called F-actin. F-actin. one single individual circle is called G-actin. On top of this, attached, almost looking like Mickey Mouse ears with a face, attached up there we have troponin. Now I'm going to blow up troponin a little bit so we can see it better. Troponin will look like this. Face, 2 ears, right? With troponin, we have troponin T, troponin I, and troponin C. Troponin T has an affinity and connection with tryptomyosin. Troponin I has an affinity and binding to G-actin. With actin. F-actin, but it's actin. Okay? Then C has a high affinity to calcium. And so we call it troponin C. Now, before I explain any more, one last thing I need to do on here is explain this part here. Attached to titin is a structure called myosin. Okay, now myosin has these pieces coming off of it called myosin heads. They look like this. Now I'm going to blow this up and show it a little bit bigger as well. And what we see with these myosin heads, or they have the myosin head, okay, and then they have the neck, and this is the tail, and this area here is called the heavy chain, and then we have these areas here on the sides, and these are called our light chains. Okay, light chains. So, what ends up happening is we have this calcium here, troponin C, that will bind to here. Now, troponin C has four affinity zones. So we have 2 high affinity and 2 low affinity binding sites for the calcium. Okay? So what ends up happening is we have this reaction that causes calcium to come out of the sarcoplasmic reticulum. Now with skeletal muscle, we're only going to have calcium coming from this supply that actually causes muscle contraction. What ends up happening is we end up having this calcium come flooding out, and it comes over to troponin C. And troponin C has those high affinity binding sites, 2 high and 2 low, and so our calcium binds to troponin C. When calcium binds to troponin C, it causes something really important to happen. There's a structure right here that runs like this, and it runs on top of the G-actin active sites covering them, and it's called tropomyosin, which is right here. So this is our tropomyosin. What happens is when calcium binds to the troponin C part of troponin, it actually causes a conformational shift moving tropomyosin out of the way, revealing the G-actin active sites. When the G-actin active sites are then open, it allows this myosin head right here, you see that, to come up and bind with that G-actin active site. And so that's the next part that I'm going to draw out here, is actual muscle contraction with the relationship between myosin and the G-actin active site. So what ends up happening is we have, if this were part of the Z-disc and this part here was nebulin, and then we had circles here being F-actin, and then individual ones with active sites being G-actin active sites. What we would notice here is we would have our myosin head in what we call a 90-degree angle, and this would be known as, it is in a cocked position. Cocked position. What ends up happening is this has a phosphate bound to it and a ADP. If troponin C bound to calcium can bind to calcium, it's going to remove the tropomyosin, like we were saying. So now the active site is open, okay? So now we're going to see the head actually come up and bind. That's what we would see here in this figure. We're going to see the head come up and actually bind to the G-actin active site. Well, that binding actually causes us, if we input calcium, what that does is it's going to actually be a lot of energy, and it's going to break this bond right here in order to continue going with contraction. So if we break the bond, then we're going to kick off this phosphate. In doing so, we're also going to release our ADP. Doing such, releasing the phosphate, causes our myosin body to uncock and to pull down. When it pulls down, this is what it would look like. We would have it like this, here and here. Now, to get it to release from the G-actin active site, we're going to add in ATP. When we add in ATP, it releases, but it stays in the uncocked position. And as it's in uncocked position, so it's in the uncocked position. So what we want to do is ATP's been added. Now we're going to do hydrolysis. And when we do hydrolysis, we're going to cut ATP in half into ADP and phosphate, and then we're going to recock it, and it's ready to attach again. Okay? So this is how muscle contraction works. I'll explain it really quick again. Calcium comes in, binds to troponin C. Troponin C, when it binds, causes a conformational shift. It moves tropomyosin out of the way. When tropomyosin's moved out of the way, we end up having the ability for the myosin head to attach. If myosin head has already been hydrolyzed and we have ADP and a phosphate attached to it, then myosin will bind to the G-actin active site because it's no longer blocked by tropomyosin. When it binds, we end up having too much energy and it releases in breaking the bond with the phosphate. We kick the phosphate off. In doing so, we kick off the ADP, but it's really the phosphate that causes the myosin head to pull. And we call that the power stroke. That's something I forgot to say. We call that the power stroke. This is called power stroke. Power stroke. That's called the power stroke, pulling it. So myosin attaches, phosphate kicks off, myosin pulls. Then we want to release it. We add ATP, we release. ATP has been added, now we hydrolyze it and we recock and we're ready to bind. Then we kick phosphate off and we pull. And we add ADP, ATP, we let go, it releases. After ATP has been released, or bound to it to release it, ATP is hydrolyzed. We cut it in half, phosphate, ADP, and it pulls back up and it's ready to bind. And that's the process of muscle contraction. Now, when we talk about relaxation, we talk about wanting to get rid of the calcium. Really briefly, we can pull it back into the sarcoplasmic reticulum via something called a Circa pump. So we would use the Circa pump. When we use the Circa pump, this is going to be primary active transport. And we're going to be pumping calcium ions against their concentration gradient using ATP at high expenditure to get calcium back into the cell. And this is where we're going to notice muscle contraction occurring, is within this full system right here. Okay? Now, let's get moving on to the next section. Okay, I want to move on a little bit. Let's see if I can put this right here, if this will work with this board over here. Is that holding up? How's that? That's pretty good, huh? Okay, so what we have right here, I want to explain one little thing, and that's called muscle cell energy type. Now, we know in cells, for what we just explained with muscle contraction to occur, we need to have ATP. There's multiple ways we can get ATP. We know that ATP is the power or the energy source of energy for our muscle contraction. And why are we talking about this? Because we're talking about smooth muscle, okay? With smooth muscle, we need ATP for contraction to occur. So what ends up happening is we have a couple. We have, number one, we have cytosolic ATP. And cytosolic ATP is just stuff that floats in the cytoplasm. It could be a lot, could be a little bit. And so we don't really trust it, not the best. This is no O2, no oxygen needed for this kind of ATP production. It's just sitting in the cytoplasm. And what ends up happening is this lasts 1 to 5 seconds for muscle contraction. The second thing is creatine phosphate. This is done by an enzyme called creatine kinase. Also uses no O2 and lasts about 10 seconds. Not the best. Creatine phosphate. Then we have number 3, source of energy, is called the glycolysis. Now, in glycolysis, we know that we use glucose, carbs, carbohydrates. You got fructose, glucose, all these other ones, ribose. You could have mannose, maltose, which is a disaccharide. You can have all these things converted, broken down into what we call glucose, which then gets fed into glycolysis. We know that we net 2 ATP. And so just by glycolysis, we can produce ATP to feed the cell. This lasts about 1 minute. Okay? But the best way is cellular respiration. And with cellular respiration, we use oxygen. Okay? We produce a lot of ATP. And we mainly want to use fatty acids. And how this works is through beta oxidation, right? Fatty acids through beta oxidation get fed into the Krebs cycle, citric acid cycle, which then get fed to the electron transport chain, which actually pass on all of these electrons to then produce ATP. And this lasts for hours of muscle contraction, okay? So cellular respiration is the best way for us to produce the ATP that we need for our muscles to contract, okay? So let me show that one more time here on the board. Perfect. All right, let's get back to where we were. Okay, so for this next part, I've already done neuromuscular junction, right? We've already done muscle cell energy types, okay? We talked about ATP in the cytosol. We talked about cytosolic ATP, creatine phosphate. We talked about glycolysis producing ATP, and then we talked about cellular respiration with metabolism through glycolysis. But mainly beta oxidation with fatty acids is where you're going to get the most energy in terms of ATP to feed those muscle cells so they can fire through the cross-bridge cycle. So this is called the cross-bridge cycle. And I can't believe I didn't label that on that last part of the video, okay? So what I'm gonna do is I'm gonna explain a couple parts of the smooth muscle, okay? We have 3 parts labeled up here. The first one is slow type 1. The second one is fast type 2A. And the third one is fast type 2B, or we call it X, okay? There's going to be a couple things about each of these that I want to explain. I'll draw a couple things out as well. When we talk about smooth muscle, what we're talking about is muscle that's cardiac muscle, right? We're talking about intestinal muscle. We're talking about smooth muscle with blood vessels. These muscles that need to do things that are different than skeletal muscle, okay? So, actually, let me think here. Smooth muscle. Yeah, that's what I'm thinking. Well, I don't know if these are smooth muscle. I think these might be muscle fiber types. Yeah, I don't think these are smooth muscles. So I don't think we're talking about the cardiac muscle stuff yet. This is just muscle fiber types right now. Okay, so kind of scratch what I said there for a second. This is muscle fiber types, okay? So our first muscle fiber type, I'm going to explain a couple things. So structure, metabolic function, and then their actual function. So structure, metabolism, function, structure, metabolism, function, okay? So for these first one, the slow type 1 muscle fibers, what we know about these is they have high blood supply, right? They have high blood vessel blood supply going to them. And that's because in diameter is very small. These slow muscle types, type 1 muscles fibers. They're very small in diameter, so the blood vessels increase blood supply therefore they have a red color to them. And because they have a red color to them, they stand out different than some of these other cells. We also know that the mitochondria in them is very high. They have high amounts of this mitochondria going through them. Therefore, we have high ATP production. Now, if we know that the diameter is small, that's what leads to that high blood supply and the red coloration, okay? And so in terms of energy, this takes aerobic cellular respiration. And we know that it takes aerobic cellular respiration using O2, so it uses triglycerides and beta oxidation, right? So its function is going to be slow muscle contraction. Slow muscle contraction. Because it has slow muscle contraction, we're going to have less ATPase working. Decrease ATPase, right? So if we have slow muscle contraction, then we're going to decrease myosin ATPase activity. When we decrease the myosin ATPase activity, this creates increase in long contraction. Okay, if we have longer contraction, then we have a decrease of the circopump activity. Okay, with decrease in circopump activity, we become fatigue resistance. And this is known as marathon muscles endurance. Okay, I'll kind of explain a little bit more about how that works, all right? Now, our type 2. Type 2, these are going to have so in terms of their diameter, we'll start with that. They're going to be a little bit higher. So mid increase in diameter. Okay? They're going to mildly increase in their diameter. Now, if they increase in their diameter, then we know that the capillary density is going to increase as well. Just a little bit, but it decreases somewhat. So this is kind of like a pinkish red because capillary amount of blood vessels going through are not as high. So mid capillary density. Okay, so if we have mid capillary density, then they're kind of that light red with the myoglobin. Almost like a pinkish, right? But they still have that red tint to them. Now, they have mid amounts of mitochondria as well. So if we had 5 over there, maybe we would just say they have, 3 over here. And we have this, decrease a little bit. Mid amount of mitochondria, okay? So we have this medium amount of mitochondria that exists within these fast twitch muscle fibers. So basically what these are, because I scratched everything I said at the beginning of this section with them being, smooth muscle stuff, cardiac, that's not what they are. They're actually the oh gosh, what's it called? Extrafusal muscles. So these muscle fibers, right? So when I was talking about it over here, you have skeletal muscle, the endomysium. Then you have these muscle fibers called fascicles. Then at that lower level, you would have muscle fibers, and that's what these are, these different types of muscle fibers. So you have these slow, kind of thinner ones, and then you have these medium ones that are a little bit thicker in diameter, and so we see everything else kind of drop a little bit with those. So we have that decrease in mitochondria, so we have kind of a little bit of a decrease in O2, right? Because mitochondria is going to produce a lot of ATP. And so these can do aerobic and anaerobic. So these can do anaerobic and aerobic functions, okay, for metabolism, and where they get their energy through glycolysis. So the function would be increased contractile speed. Because we do that, we would increase ATPase. Yeah, so we'd increase myosin, ATPase function. We would also increase, I believe, the Circa pump. We'd increase our Circa pump activity. And we would also increase, we would decrease contraction length, shorter contraction length. And if we increase larger so this would be moderate fatigue. So this would be walking or sprinting. Okay. Now, based off of what we know on structures on these other things, this would probably be the highest in diameter, thickest. Because that could be very decreased in blood vessel supply. So it would appear white, few hemoglobin, or what we call myoglobin. It would appear white in color. Diameter would be large, that would be down, and low mitochondria. Right, we would have low mitochondria. Well, low mitochondria, not low, no, but low, so maybe just we'd put 1 in here like that, okay? For metabolism, this thing would be using anaerobic glycolytic processes. So this could use creatine phosphate, or it can use glycolysis. And we use anaerobic pathway processes. Okay? So we use anaerobic pathway processes through glycolysis. So duration of contraction would be even slower. which would mean that we would have increased circuit pump even more, uptake of the calcium, and we would have increased ATPase, myosin head activity. So then we would have fast contraction, and we would have increased easily fatigued. So this would be easily fatigued. Okay? So let's run through this one more time. Muscle fiber types. We have slow type 1 muscle fiber types. We have fast type 2A muscle fiber types. We have fast type 2B or X muscle fiber types. How they work is they are the extrafusal muscle fibers. So they're the little thin fibers that have all of the actin, myosin, nebulin, and the titin all within it. Okay? So what ends up happening is you have your structure, you have a decrease in diameter with the slow type muscle 1 fibers. Because they decrease in diameter, their blood supply is going to increase. If their blood supply increases, they're going to be red. They use metabolic pathway of aerobic cellular respiration through oxidation. Then their function is slow muscle contraction. Okay? What that means is we have decrease in ATPase myosin heads activating and binding to the G-actin active sites. We have long contractions, which means we have decrease in circa pump activity. This fatigue is resistant. These muscle fibers are resistant to fatigue, so that's why they're marathon and endurance cells. Then we have type 2 fast type 2A muscle fibers. These are going to have kind of the medium of everything. Oh, and I even said increase in mitochondria right here. Okay, so now we have structure. We have like a middle range in diameter, medium capillary blood supply. We have a medium reddish color. We have that light red. Then we have a decrease in mitochondria activity, and we have a decrease in O2 function. So our metabolic pathway is going to be aerobic respiration and anaerobic respiration. Our function is to increase contractile speed. It increases myosin ATPase function, increases circuit pump activity, and decreases shorter contraction length, moderately affected by fatigue. Walking, sprinting, probably the toughest for it, or the best way to explain what those muscles are used for. The last one are fast type 2B muscle fiber types. Those are going to have an increased, biggest diameter, smallest blood supply. They're going to be white because they don't have that high blood supply that makes them red under the microscope. Then mitochondria is going to be low in activity within these muscle fiber types. Then for function, they use creatine phosphate as a form of ATP for their metabolic function, and then glycolysis anaerobic pathway processes using glucose with glycolysis produce ATP. The duration of these muscles for their contraction is very low. Because their contraction is very low, we have a circuit pump. Circuit pump is going to decrease these. The circuit pump for calcium will increase. If the circuit pump for calcium increases, we also know that there's an increase in ATPase with the myosin heads activity, and we have an increase in fast contraction and increase in them being easily fatigued. So these are high power movements. And so we would say that this would be weight lifting. Okay, weightlifting. Perfect. Okay, so I'm gonna add in a couple things here. Okay. All right, let's see where we at here. Perfect. All right. So there's one other thing I wanted to talk about in terms of when we were talking about muscle fiber types. Okay. Again, we know the muscle fiber types are slow type 1 muscle fiber types, slow or fast type 2A muscle fiber types, and fast type 2B muscle fiber types. Okay. When we talked about them, we know that the type 1 muscle fiber types were very slow or didn't even fatigue, okay? The type 2 muscle fiber types, they were moderately affected by fatigue. The type 2A muscle fiber types were. The type 2B or X muscle fiber types were very affected by fatigue, and so they didn't last very long, okay? So what we know about fatigue is this. And this is kind of a hard concept for physiologists to understand at this time, but we have psychological fatigue. Okay, and we have physiological fatigue. Okay, what ends up happening is this is mind. Okay, this is in your head. Physiological or psychological fatigue. We tell ourselves, oh, I'm tired, I can't do this. That's psychological fatigue. Physiological fatigue, there's two types of physiological fatigue. Okay, we have low frequency and high frequency. Okay, low frequency, high frequency fatigue. Low frequency is they detect that you have low levels of ATP. Okay, so to correct this type of fatigue, what we end up, you're running down low on ATP. We want to shut off the circuit pump. Shut down the circuit pump. But we don't want to leave a ton of calcium in the membrane. In the sarcoplasm. So what we end up doing is we use magnesium and we block RYR. Do we remember where RYR is? RYR is right here. And we put Mg plus right there. And we block it. If we block that, then calcium can't get in. So if calcium can't get in, then we stop contraction and we don't need to use the circuit pump as much to pull calcium in. So we want to shut down the circuit pump, and we do that by shutting down calcium coming into the cell, thus binding to troponin C and leading to no contraction. So it wouldn't bind to troponin C and it wouldn't lead to, and there would be no contraction following that, okay? That's low frequency fatigue, physiological fatigue. High frequency physiological fatigue is we have this potassium buildup in the T-tubules, okay? Because we have this action potential happening, following behind it are the calcium ion channels to cause depolarization. As we come around, this is a little sink, and so we end up getting tons and tons of these protons, or these potassium ions down there in the T-tubules. And so if you have this high concentration building up, then right here within the membrane, we know potassium wants to leave, but it goes from high to low. But if these have a hard time diffusing out of the T-tubule, groove, indentation, then you're gonna have high concentration of calcium or potassium and low concentration of potassium on the inside. And so concentration gradient is going to want to come in. If we can't get rid of potassium from the inside, our cells stay at a net positive charge, and so we have a continued action potential. DHP stays open, RYR is open, and calcium continues to flow into the cell. So to correct this, because we want it to become more negative, we have these chlorine leak channels and chlorine voltage gated channels. Okay, so we have leak channel and chlorine voltage gated channels. So if these channels can open up, then we allow that chlorine to come into the cell, because chlorine's high on the outside, it wants to come inside, then we make it more negative. And we counteract that problem with fatigue that builds up with calcium right there, okay? So those are two ways that we deal with fatigue. All right, so next section with smooth muscle. I went through 3 types. I want to go through contraction, and I already went through fatigue. Sensory receptors. Okay, so I want to go through fatigue, or I already did fatigue. I want to go through muscle contraction now. Okay, let's move down. Okay, so for this last kind of section part here, hopefully I can see that. We can see that. I want to go over muscle contraction, or smooth muscle. Okay, smooth muscle. So what do we know about smooth muscle? Well, we know that smooth muscle are going to be on our lungs, blood vessels, heart, various organs, digestive organs, urine tubes, those kind of things. These like involuntary muscles that we use that we don't, we're not controlling with our head, if that makes sense. Why does this look so weird? All right, how's that? Good? Can I even read that? All right. So with smooth muscle, what we know is that we have different ways of getting calcium to fire. So these don't have troponin. Okay, so there's no troponin C, and they don't have a sarcomere. And they're not anything like the skeletal muscle, okay? There's no T-tubules. No T-tubules, no, so I believe most of these are activated by stretch, okay? So we need an increase in calcium to occur in order for these to fire, okay? So what ends up happening is you could have a gate like this that's open up, and we can allow calcium to come in. We would call this a voltage gated ion channel. So if this is not using the same kind of pathway with T-tubules and action potential firing like skeletal muscle, then how is calcium getting in, and then what is it going to do once it gets inside? Well, number one, it can use these voltage gated ion channels, and we can have this positive charge come inside and then we have calcium ions flowing within. We can also have more of a chemical message. These would be ligand channels. These would be so neurotransmitters, or we could have oh gosh, what other chemicals do we have? Sorry, my brain's dying right now. We could have let's see, hormones or neurotransmitters or chemical receptors. So neurotransmitters, like I was saying, or we could have hormones. Hormones right there, okay? If we have hormones, so we would have hormones. And so what these do is they cause this cascade, right, of IP3. And it's a G coupled protein, GCP. Receptor. GCPR. Right, G-coupled protein receptor. And so what these all do is they go to the sarcoplasmic reticulum. Okay. So they go to this sarcoplasmic reticulum. In the sarcoplasmic reticulum, we have high concentration of calcium, right? IP3 can bind RYR. When it binds to RYR, opens it up, allows calcium to flow out. We also have calcium-induced channels. These calcium-induced channels somehow change positive charge. They're probably calcium voltage gated channels within the sarcoplasmic reticulum, and they allow that calcium to flow off and out. The important news is calcium comes from both the ICF and the ECF in smooth muscle. Okay, if calcium's coming from the ICF and the ECF in smooth muscle, then what this means is we have a different way of muscle contraction. So we're gonna have two different ways. One is called phasic, and the other one is tonic. Okay, phasic and tonic. So let's start with phasic first. Phasic is also known as rhythmic. Okay, and tonic is also known as, sustained contractions. Okay, so if we have phasic and tonic, we have these sustained contractions, we have rhythmic. What we end up having is this. We have the G-actin, okay, right here. We call that the actin, and this is myosin here in this cocked position. It has ADP, and it has a phosphate on the neck. What ends up happening is we come right here, and in order for this to bind, we actually add in calcium and we add in MLC, okay? which is called myosin light chain kinase, and we add in ATP. Over here, we have our myosin light chain, or our structure over here called calmodulin. Now, calmodulin is a structure that has 4 affinity binding sites for calcium. So you need 4 to come over here and bind to this. Once all 4 bind to this, let me make sure I'm doing this right. Let's see, MLCK, all 4 bind to it. Yeah. Once all 4 bind to it, then it causes a conformational shift within MLCK. Or sorry, within calmodulin. This then binds to MLCK. When this binds to MLCK, MLCK then comes over with ATP and adds a phosphate onto this myosin. Okay, with myosin, or with this phosphate added on, we now can attach. So what we see is we see ADP here, and we see these phosphates, it attaches, okay? Because it then attaches, we now have now that it attaches, so this phosphate is going to attach when, or the myosin attaches. When the myosin attaches, it's going to cleave off one of these phosphates here. So we draw our arrow here, and we see that when this attaches, we now go into our uncocked state. When it goes into the uncocked state, we still have one phosphate here, and we got rid of ADP as well, okay? Now we come up, and right here we're going to add on ATP so we can unbind myosin head. So we stay cocked down, we still have the phosphate. We have ATP. And ATP. And we hydrolyze it. When we hydrolyze it, this goes up. And now we have 2 phosphates and our ADP. And we can cut right over to here and bind. We're right here in the cycle. We can skip this step right here. Okay? We can skip that. If we want to get rid of this phosphate and end the cycle, though, we use MLCP. And MLCP will come in and cleave off this phosphate. This then becomes the relaxation phase. Okay, MLCP comes in, takes that off. Now, if we want to inhibit and block MLCP, we use Rho kinase. And Rho kinase will come in and add a phosphate onto the enzyme. And if we add the phosphate onto the enzyme here like that, then it's not going to want to cleave that off. So if we increase Rho kinase, eventually it comes off and it will cleave off the phosphate from the phasic section. Okay? Now, a little bit the same with this next one with tonic, but we're going to have a lag phase. Okay? So what ends up happening with tonic is we're going to have the same thing with our structure here. So we're going to have our myosin head, neck, we're going to have ADP, and we're going to have our phosphate attached here, okay? And then we're going to be attaching to this actin here to pull for the power stroke. This part here, when it pulled down, when we got rid of that phosphate, this was our power stroke. Okay. So what ends up happening is right here, we're gonna have this myosin head bind. As this myosin head wants to bind, we gotta get it ready. So this is in the cocked state. In order to get it ready, we're gonna add in calcium. We're gonna add in ATP, and we're gonna add in MLCK. Myosin light chain kinase. This is myosin light chain phosphatase right here. Okay, so we add all these things in. When we do, we actually cause the same thing to happen with MLCK, where it's going to add a phosphate onto the myosin head. So we're going to add a phosphate on. And we have a phosphate over here. Because of that, it will then allow us to bind myosin there. When we do, or with actin, when we do bind with that, we end up cleaving off that phosphate. So we're in the uncocked position. When we're in the uncocked position, what ends up happening is we get rid of ADP, we get rid of the phosphate, and MLCP, if it's in high concentration, in this case with tonic, it's going to come in early and it's going to cleave off the other phosphate here. And when it does, then we're going to release another phosphate. So we're here. To have it release, we want to add in ATP. So we release, but we're still in the cocked position. So we're still in the cocked position. And we have ATP on here. Then it gets really weird and it goes through this lag phase. And this lag phase is caused because of this phosphate getting cleaved off early. It makes it weird with the binding of ATP. But eventually it hydrolyzes and we start the whole process all over again. Oh, it's not called the lag phase. It's called the latch phase. It's a long time. So in this case, we have sustained contraction without extra use of ATP. Okay? Perfect. So we have sustained contraction without the use of ATP. And this would be like your anal sphincter and those things, like your bladder, holding those, staying contracted for a long period of time. They have this way of getting into the lag phase. So what ends up happening, myosin comes in. It's got ATP, ADP, and a phosphate. It's already been hydrolyzed. We're going to go through a phase. MLCK is going to come in. MLCK is going to, we have calmodulin. It's going to take the 4 calciums, bind to those affinity sites. It's going to then bind or then change some things or attach to the enzyme, which is going to be myosin light chain kinase, MLCK. Then MLCK will take an ATP, and that kinase will phosphorylate and add a phosphate to the myosin neck or head. And so then on the myosin neck, we have an extra phosphate. So then we go through the cycle. As we go to the next step, it gets snipped off with, well, because then it's ready to attach. It attaches. Once it attaches, energy releases the phosphate and the ADP. When the phosphate and ADP get released, then we have the ATP, or not ATP, we have the MLCP come in. Myosin light chain phosphatase will come in, snip off the that phosphate. So then now when ADP attaches to release it, because we did our power stroke when we got rid of that phosphate, we did the power stroke. Now when ATP comes in, it's all messed up and weird. So we hit this latch phase. And in the latch phase, we stay contracted for a long period of time. These contractions can last for a long time. Okay? Okay. I'm going to go over a little bit of reflexes. So muscle reflexes. So with muscle reflexes, we know that we have these muscle and then get attached to these tendons. They're attached to the bone, right? These are called extrafusal muscle fibers. And then within here, we have this pocket within here, right here, and these are called our intrafusal muscle fibers. Okay, so these are involuntary, and these are often known as our proprioceptors. So they have the ability to sense where we're at in time without us, really knowing what's going on. In terms of like if our eyes were closed, we'd still be able to know how we're doing this, okay? We would still know where our hand is with our eyes closed. Sorry, I'm trying to get this right. Okay, so muscle reflexes. There's 2 types of these. There's nuclear bag and nuclear chain. Okay, nuclear bag, nuclear chain. How the nuclear bag look is like this. be the nucleus. And then you would have your sarcomere here, right? And then down here for the chain, it'd be like this. Okay. These ones have receptors that come in and come across like that. Okay, these are called gamma. These up here are gamma receptors. Gamma receptors. Gamma receptors. Okay, these are type 1 fibers. These are type 2 fibers. So in terms of knowing a stimulus, if this was, muscle on top of the leg and this was muscle on the bottom of the leg, so this was hamstrings, and these were quads, then how we would have this is like this. We would have our neuromuscular junction, dorsal, sensory. This would be your type 1. And type 2. Receptors coming through. And they would come from this thing here. And then they would come here and we would have this lateral horn. Like that. And they would come and they would synapse, bifurcate. And this one does that for negative inhibition. This one would come down. Positive excitation. Then they go back. And positive would come to the front if we did a little tap on our patella with a mallet. Boop, boop, boop. That would cause it to stretch. They're determined based on stretching. So it would cause stretch, they would pull. And that would be how those would work. And these would come here to do inhibitory and relax the other side. And this is called their interneuron. The same is with Golgi tendons, except for this here. With Golgi tendons, this is type 1B. And if this was the tendon, that's where Golgi tendon organs would be. Right? And they're proprioceptors. And so their main thing is to detect tension. And so how they work is if you get too much tension on the tendon pulling on the bone, then it sends an inhibitory to your contracting muscle and excitatory to your antagonist muscle, because it wants to relieve pressure or tension that it's detecting on the tendon. And so how that one works is they would have their innervation here and they would come down this lateral horn to the dorsal area. And so what's crazy is they synapse here in the spinal cord. They don't synapse up into the brain or the CNS. Well, that part of the CNS, they're in the CNS part with the spinal cord right here. Okay. And so that is going to be how Golgi tendon organs work through stretch. Withdrawal reflex, just they work simultaneously with the stretch reflex. And then that's muscle spindles. Okay, that's everything.